Good afternoon, everybody. I would like to welcome you to the fifth webinar on, of the Quality Webinar Series organized by EATRIS. This webinar is on manufacturing of ATMPs. My name is Merel Ax. I work for the Eureka Career Center of KU Leuven, and I am co-hosting this webinar today together with EATRIS in the Netherlands. Today, we are very pleased to have Professor Dr. Frank Luyten and Dr. Kristel Van Landuyt joining us for this webinar, which is the second in a series of 3 technical webinars on ATMPs in the Advanced Educational Program about the exciting world of advanced therapy medicinal products. Advance is an innovative and focused learning program for future developers of advanced therapy medicinal products, ATMPs. It consists of 4 e-learning modules, 2 cycles of 7 webinars, and 2 face-to-face 5-day workshops. It's for PhD students, early postdocs, and doctors in trainings. Here you can see the advanced program partners, EATRIS and Elevate in the Netherlands, I3H of the ULB and KU Leuven in Belgium, the University of Ljubljana, Istituto Superiore di Sanità in Rome, and Takis Biotech Company in Italy. Please note that this session is being recorded. The recording will be made available at the end of this session on the website of EATRIS. If you have any questions during this webinar, please type them in the question box and press send. If you would have any follow-up questions after this session, you can always send them to advance@eatris.eu. If you want to have a better view on the PowerPoint presentation, you can press the orange mark on the top left-hand corner of the control panel. And then I'm very happy to introduce our 2 speakers of today. Professor Dr. Frank Luyten is a full professor at the Faculty of Medicine, KU Leuven. He's a chairman of the Division of Rheumatology at the University Hospitals of Leuven. He is director of the Skeletal Biology and Engineering Research Center, KU Leuven, and director of the Leuven Research and Development Division, Prometheus Skeleton Tissue Engineering Leuven. He was founder and member of Board of Directors of TiGenix and member of Board of Director of Pharmaceutical. He is Medical Scientific Director of Regenerative Medicine Across Borders, Netherlands, Flanders. The second presenter of today is Kristel Van Landuyt, and she's a staff member of the Biobanking Department of the University Hospitals Leuven and a practicing rheumatologist. They will both give a presentation, and at the end we have reserved time for questions and answers. The questions and answers will be moderated by Professor Dr. Isabel Huis, who is a pharmacist by training with a PhD in pharmaceutical sciences, toxicology, and further specializations in regulations on medicinal product development at KU Leuven, including intellectual property rights. I'm very happy to give the floor now to Dr. Kristel Van Landuyt.
So, dear students and colleagues, welcome to this presentation. So, first I will present a rather theoretical part, including important aspects like banking and traceability of cell-based products. Then Professor Luyten will present a real-life application of cell-based ATMP, namely ChondroSelect. This will be followed by answering your questions and having a discussion. ATMPs are a class of biological medicinal products that is defined by law. In 2007, when the new regulation on ATMPs was published, the European Medicines Agency officially launched this type of product as an entity on its website. From that moment on until now, the application of the ATMP regulations has been dynamic, as is the field of ATMP development and manufacturing itself. So it's a good idea to keep updated with regard to this website. So the legal definition of ATMP includes gene therapy, somatic cell therapy, and tissue engineered products. During this presentation, we will rather focus on cell-based products, and so in the application specifically even on a tissue engineered product. So ATMP manufacturing has some aspects in common with other pharmaceutical industries, but also shows important differences. We want to take the specificities of ATMP manufacturing as a starting point to explain the many challenges. There is a series of commonalities with medicinal products in general, including the manufacturing in GMP licensed facility, a clearly defined production process, the need for quality management systems, and the need for appropriate logistical organization. GMP, or Good Manufacturing Practice, is defined by law and applies to all pharmaceutical industries. It is regulated in general, but for ATMPs separately in the Udralex guidelines on GMP specific to ATMPs in force since May 2018. National competent authorities inspect the facilities and can grant GMP license. The interpretation of ATMP-specific GMP requirements can be different between European countries. ATMPs are often investigational MPs, and during development, an important effort has to be delivered to translate the preclinical phase into a GMP-compliant production. This development can take place in academic or in more commercial environments. Every step of the manufacturing process has to be defined, validated, and described in standard operating procedures and batch documents, including use of reagents, materials and equipment, formulations, packaging, release, storage, distribution, and all aspects of quality assurance, which have to be integrated in the process. If possible, automated steps will be implemented if there is a benefit above manual processing. At the left, you see the two aspects, like being an upstream process and there is a downstream process. At the right, there is a commercial example of the production of biological products and the complex flow of process. Quality. Quality has to be assured in every possible aspect of by a pharmaceutical quality system. This is a sum of arrangements made with the objective of ensuring that medicinal products are of the quality required for their intended use. Compliance with GMP is an essential part of the quality system. The Udralex mentions that in particular should be ensured training of personnel, suitable premises and equipment and their maintenance, an adequate documentation system, consistent production, independency of the quality control system, prospective change control, handling of deviations and quality defects, and systems to ensure traceability of the ATMPs and their starting and critical raw materials. The European Directorate for the Quality of Medicines issues guidelines, for instance in the form of monographs, covering all aspects that impact on quality and that have to be followed to comply with legal requirements. Even with the best quality system, the manufacturer cannot function on its own, but needs a suitable supply chain and logistical arrangements with vendors and with users. For instance, all starting and raw materials have to be timely available. The production, the stock and distribution are coordinated to be compatible with demand, indications for use of the products, and with the location of the users. The manufacturer has data about the place of its products into the market of pharmaceutical products and about cost effectiveness. Even while looking at commonalities, it becomes clear that already for each aspect, specificities of ATMPs pop up. We will now go on by focusing on the specific aspects, divided in first, the starting materials, as we already said, tissues and cells, aseptic manufacturing, storage and shelf life, transport and handling, and then, as we already said, the real-life case presented by Professor Luyten. So how do we start? Starting materials in ATMPs are typically human tissues and cells as it concerned cell-based therapies, besides other raw materials of biological origin. This leads to an inherent variability. The legal definition of tissues and cells excludes blood products and organs, which are both separately regulated. The law defines a tissue establishment as an organization which will perform certain steps in the chain from donor to patient in a qualified manner. First, the task of the tissue establishment is to perform a compliant and qualified donor selection, procurement, and testing of the material. The tissues and cells can be handed to a manufacturer if the material fulfills the release criteria. After production by the manufacturer, the ATMP is again tested, stored, released, and then distributed to the user for clinical application. The fact that the material is handed between organizations required provisions for traceability. If the material is not used for ATMP production, the tissues or cells can be processed and released in another way. Traceability is the ability to identify all biomaterials from donor to receptor in all stages of the process and at every moment. It applies to the tissues and cells and to all products that come into contact with them. Why is it useful? It is needed because each starting material bears unique risk factors linked with the donor, and because the processing itself leads to a number of risks for the product, for instance, contamination, and because tissues and cells are, by law and by ethical standards, not to become replaceable trading goods, but preserve their status as a unique human gift, so are therefore handled through a principle called custodianship. Where do the materials come from? Well, they come from a donor, so they can be autologous. That means that the donor and the patient who receives the material are the same person, for instance, with CAR T cells. However, if it is not the same person, so in allogeneic tissue and cell therapies, the donor of the material is not a receiver or patient. The donation is a well-described process which is preceded by an informed consent for a living donor. In case of a deceased donor, it can be a presumed consent, like an opt-out system also. All donated materials are coded. In most cases, it is not allowed to give information about the donor to the patient. So the ATMP manufacturer would concentrate on the development of the product itself. Therefore, typically, a tissue establishment receives the donated material and provides it to the ATMP manufacturer in the framework of an agreement. Because of short timelines, it is possible that the material will be only very shortly in the tissue establishment, and even there may not enough time to await all test results. The agreement has to describe at least specifications for starting material, the transfer of information and test results, and on donor health information. Perhaps also the possibility of audits. Here below, you can see some examples of starting materials like bone marrow, fat tissue, umbilical cord, or placenta. It is of note that tissue establishments, in many cases, process the donated tissues themselves without substantial manipulation and release them directly for therapeutic use in patients, so for tissue transplantation. This practice is performed under so-called GTP, or Good Tissue Practice Guidelines, which are close to but different from GMP regulations. So it is a different chain not to be confused with ATMPs and applies, for instance, on hematopoietic stem cell transplantation, on bones and tendons for orthopedic applications, on corneal transplantation, amniotic membrane use in dermatology or ophthalmology, and on cardiac valve transplantation or skin. For a part of tissue transplantations, material is not viable and can be irradiated, for instance, processed bone. However, ATMPs are in many cases a living therapy, that cannot be terminally sterilized. So aseptic manufacturing is therefore strictly necessary, meaning a process that prevents microbial contamination. It entails an appropriate environmental cleanliness level, provisions for the introduction of the materials in the clean area, and validation of the aseptic process and of the used sterilization method. Also, the packaging has to be performed in aseptic conditions. Then the finished product has to be stored under adequate conditions to preserve quality and prevent mix-ups. Dependent on the product, the storage, temperature, and humidity conditions may be different. For longer term, many cell therapies are stored in the vapor phase of liquid nitrogen. Liquid nitrogen is extensively used for cell storage. Many solutions are commercially available, and here, apart from monitoring of the storage quality, the safety of the personnel is of important concern. Continuous monitoring of at least the temperature is mandatory. This is a critical quality parameter. In general, finished products are held separately in quarantine until their release. Physical quarantine may not be always possible due to a very short shelf life. The shelf life of ATMPs is variable, depends on the choice of the package and the storage condition, mainly temperature. Often these shelf lives are short for cell therapies, requiring adapting strategies for logistics and dealing with results of release testing. The transport of ATMPs also has to be adapted to its storage conditions and shelf life. The location of the manufacturing site, the cooperation with transport companies, and the choice of control systems can play a role in the quality, feasibility, and cost of the transport. A dry shipper is typically used to transport materials at temperature lower than 150 degrees Celsius, minus 150 degrees Celsius. This device absorbs the liquid nitrogen so that it does not contain any liquid anymore. It can thus be transported with caution, but as a non-hazardous item. For other transport temperatures, and recently also for lower than minus 150, a smart shipper can be a good solution, with extensive real-time online tracking possibilities. This is a specific commercial solution from one company dedicated to the needs of ATMPs. The system fulfills extensive traceability and documentation options. So when the ATMP is then received on the site of use, being a hospital, the preparation preceding its application to the patient is called reconstitution. This can be done outside GMP conditions, presumed that no substantial manipulation has to be performed. Reconstitution can be thawing, washing, resuspension. For cell cultivation, however, GMP conditions are always mandatory. Of course, the equipment and licenses of the hospital that applies the therapy have to be in place. So here ends my overview and the more theoretical part. Now I will hand over a speech to Professor Luyten, who will continue the presentation.
Excellent. Thank you very much. We are pretty much in time, I see. A case study. We have now the frame that was brought forward by Kristel. And now I'm giving you the example of a ATMP, actually the first cell-based ATMP that obtained central market approval by EMA here in Europe. Condor Select was a dossier and helped, in fact, to set the regulatory path in place also here in Europe. First thing, what are we talking about? This quite spectacular view of a knee of a patient. This is a patient who came to see us with a symptomatic, a painful knee. Upon arthroscopy, you could see that there was a large cartilage defect. You could also do an MRI. You don't need always to do arthroscopy to see that. This deep cartilage defect does not heal. And therefore, the ATMP has as goal to repair this cartilage defect, regenerate it, And by doing so, will help again the patient, and patient can go back to the job, can go back to life. The treatment is the injection or implantation of a chondrocyte solution under a so-called periosteal membrane or a collagen flap, as you can see here on the bottom. This is the entire procedure. It's called autologous chondrocyte implantation. So autologous, we know what that means. It's material from the patient itself. Indeed, upon the defect that has been either confirmed by arthroscopy or MRI, the patient, together with his clinician, obviously, and the surgeon, decides that let's try to do some cartilage repair. A biopsy is obtained in these patients with a special biopsy instrument, actually, removing about 200 milligrams of wet weight cartilage. That biopsy is shifted to the expansion facility, where the cells are released from the tissue by enzyme digestion. This enzymatic digestion keeps a number of cells alive and viable, and these cells then are seeded and expanded in vitro to large numbers. These large numbers are then needed to actually repair the tissue damage. This is one of the critical steps here, expanding of these cells by maintaining their potency, their biological activity. We will discuss this briefly later on. Once the cells have been obtained at the appropriate number, the cells are shipped to the clinic, and the clinician is injecting the cells under, as you can see here on the left, under a periosteal flap or a collagen membrane. And then the rehabilitation starts in the patient. Obviously, in the beginning, the cells settle at the bottom and starts to progressively make matrix, fill the defect, ultimately leading to a complete healing and regeneration. And the rehabilitation process guides this regenerative process. This is one of the things one can do in patients. Unfortunately, we cannot do this in animals. Rehabilitation. Okay, so how to make this cell product? First, you have to start from a tissue. You have to obtain the biopsy. You have to obtain the material, digest and release the cells. These cells need to be looked at from the start. Are these cells looking healthy? Are the chondrocytes in good shape? And one can do this based on a number of, for instance, molecular markers that can be analyzed in several ways, including FACS analysis. Then the few cells, in general, like 100,000 cells are released from the biopsy, are brought in vitro, and this can be under different conditions, in plastic flasks. This can also be on microcarriers, for instance. But in vitro, these cells then are expanded in proper media or culture media conditions. The cells can be followed by microscopy, for instance, looking at shape and size. Do they look normal? Do they look healthy? During this expansion process, it's always helpful to see what is going on here. One can establish growth curves, which means how fast are the cells replicating, how many passages are they undergoing in order to obtain the number of cells we'd like to have for this patient. We study then senescence also, which means are all the cells still staying happy, or is the expansion process actually negatively affecting a number of cells? Most dangerously, eventually inducing or introducing chromosomal abnormalities. So your cell product will need to be characterized also by looking at senescence and chromosomal abnormalities. This entire process needs to be ensured in terms of quality. You've heard about quality assurance system and GMP expansion. That is a key event. And I highlighted here that the biopsy is obviously a somewhat delicate part, an individual part, because all material obtained from one single patient will actually be quite different from patient to patient. That's for sure. The age of the patient will be different. The joint environment will not be the same, and so on. So you enter into an expansion process with already some level of variability, which makes it quite challenging for autologous cell products. Ultimately, it gives rise to a product, an ATMP product, that needs to be characterized. And typical aspects of characterization are shown here on this schedule. First one looks at the identity of the cells. Are they looking and smelling like chondrocytes? And one can do this by morphology, by biochemical, or other molecular markers. Also, the viability, are these cells alive, and which number of cells, how many viable cells do we actually have obtained in order to help the patient? So we have already V, viability, I, identity, P, potency, very critical here, which is a new set of criteria in place for ATMPs are the cells that you have been generating, demonstrating the biological activity and the potency relevant for the patient and relevant for the indication. So in this case, did we expand chondrocytes that maintain the capacity to make new cartilage in vivo? Because that is the intended use of these cells. They need to fill again up this cartilage defects and heal that defect. And then there is the aspect purity. And purity has to do with two aspects, and we will discuss that a little later. Finally, there is safety, and safety has to do with sterility, contaminating agents like mycoplasma, bacteria, endotoxins, other viruses, the COVID, coronaviruses, we don't want them to have in our cultures to some extent. That obviously is an important part and is also related to safety. Example here, viability, we need to define and validate viability assays. This is not always easy. New assays have been put in place that have been validated as reliable and robust assays to assess cell viability. This is quite different from what people have been used in the past for what I would call just laboratory practice. This is very different. And then the number of viable cells, how many viable cells do we need? That will depend on the size of the lesion. The larger the lesion, the more cells you will need to fill the defect. And the numbers of chondrocytes needed actually in this dossier were calculated based on the information from our colleague Ernst Hunziger in Switzerland, who calculated and enumerated the number of chondrocytes in human articular cartilage per tissue volume. So that was very, I would say, very solid and important information to come up with some cell numbers and viable cell numbers for these patients, because otherwise it would have been very difficult to define an individual patient how many cells did we really need here. So viability dosing, then the identity and potency. Identity, is the chondrocyte really still a chondrocyte? And most importantly, the potency, can these chondrocytes make cartilage in vivo? Because this is the ultimate goal of this treatment. Reimplant chondrocytes that upon implantation are restoring, are healing the joint surface defect, the articular cartilage defect. The assay that was developed for this was an ectopic assay. We were injecting these chondrocytes under the skin of a nude mice. Nude mice are mice that have an affected immune system. They are humanized in such an extent that they will not reject human cells so easily. So we injected the cells, the expanded cells under the skin into the muscle of nude mice. And when they were making, this is on the top part, when they were making high quality cartilage, we call that stable cartilage, a good quality product. When upon injection, however, the cells were not able to organize this high line cartilage, but rather a fiber cartilage, then these cells were ejected. Now, when you do this for an individual patient, you cannot every time inject all these cells into the skin of a nude mice, so you have to put markers in place, allowing you to define that indeed the cells are of high quality. And this is what you see on the right-hand side of the slide. Some of these markers were actually also published in the paper in Arthritis and Rheumatism in 2001. You need to have a number of positive markers in place, and the negative ones needs to be weak or absent. The top panel demonstrates all our positive markers are very strong. Negative markers are either absent or weak, and this was corresponding indeed with a stable cartilage formation in vivo. At the bottom part, poor cartilage, fiber cartilage made positive markers almost absent, negative markers becoming more prominent. So this was the result of about 3 years work and analysis in developing a culture expansion process that was resulting in a product that was basically, in terms of potency, also capable of making good quality high-line cartilage. The purity characterizing cells we also talked about, we had VIPP, remember, viability, identity, potency. Last point, the purity. What do we mean by purity? Number one, yeah, cells are grown in plastic flask or on microcarriers or whatever. So they may be contaminated with some materials in these containers. So we can only work with medical grade materials, and the culture and media components should be of medical grade too, which makes them in general much more expensive. At the other side, there is also something like impurities in cell populations. Indeed, one may have a contaminating cell population, and taking the biopsy, one can take some of the synovial fibroblasts or synovial fluid with it, and that may contain then fibroblasts that are not at all chondrocytes. And then the danger is that when you're expanding the cells, that actually the chondrocytes are not expanding very well, and that you are rather expanding the cell over fibroblasts, which obviously is not the case. So one can define and one can detect, for instance, by qPCR, the presence of contaminating cell populations, which you would not like to have. You would not like to implant, obviously, fibroblast or fat cells into your cartilage lesion. So this is characterization of the cell product. It took about 3 years work to put that in place, and you have to repeat this on many samples. We had about 50 samples done of 50 individual patients, every time confirming and reconfirming the robustness of the assays and the information we had on this product. The next step was then, can we go to the patient a little too early? You have to demonstrate then feasibility and potency in a larger animal model and in the joint environment, because potency was done ectopically in nude mice. Would it be the same in a cartilage defect in a relevant animal model? We chose the goat, and this was also talked about and negotiated with the regulators. The goat was a proper model, has a thickness of cartilage that is a little bit less, but I would say in the same order of the human cartilage, and the sizes of joints are getting closer to the human sizes. So in the large animal experiments, we could demonstrate that yes, we can grow the amount of cells that are necessary of good quality to repair the cartilage. And secondly, even in a longer term follow-up, as you can see here, 6 months after follow-up, the defect which was created was actually filled and healing fairly nicely in this goat. And it is with these data sets that one can consider then eventually to enter in the clinical or in the clinical trial environment and in the patient. We've learned a lot from these preclinical studies. The most important point that I mentioned here, difficulties in rehabilitation. Indeed, as I demonstrated that when you implant these chondrocytes, they need some time to start making matrix. And at that time, you want to ask your patient, please, no loading on the lesion at this point in the game. Well, this is something you cannot do in goats. This is why the results in goats, in fact, when you look back at the data, have been less convincing than we've had in a number of patients. Good. Good manufacturing practices are needed. GMP, quality assurance system. Kristel Van Landuyt already mentioned this. Before you go into the patient, you have to bring this process in place. That requires quite some investment. You can go into an exploratory trial where most of these things have not to be like 100% settled in stone, but once you obviously have a phase 3 or pivotal clinical trial, the whole process here has to be set and settled. Clinical studies, once we had the cartilaginous product, the cartilage graft, one could say, or cell suspension in place, we wanted to demonstrate that this was a successful treatment in the patient environment, in patients with large cartilage, symptomatic cartilage defects. And then we bumped into a number of challenges. Number one is, yes, in this world of surgery, many of the surgical procedures, in fact, have not been validated and demonstrated in prospective randomized trials, which brought us the comments of the regulators. What is the comparator? With what are you going to compare your new treatment with your cartilage grafting? Ultimately coming then to microfracture probably being the best comparator, but there was no 100% agreement in the field that this was the case. And since there were no prospective clinical trials for these patients, the number of patients to enroll and the statistics were much more challenging. On top of that, after all, it's also a surgical procedure because you need to open the knee or go by arthroscopy, put the membrane in place, inject the cells into the membrane. This is a surgical intervention, and surgeons need to be trained to do things like this. Then one has to discuss what are the outcome measures that are of benefit. How long are we going to follow up these patients? So a lot of these things came up, which in the field of surgical orthopedic interventions were not really yet in place or not yet understood at this point. But anyway, we went ahead and developed a study hypothesis. Study hypothesis is fairly straightforward. We believe that this cartilage grafting is a regenerative treatment, which means that if you repair this defect and you do this well, and now we can follow here the green-blue line, then the outcome over a long term will always be positive, which means it's a durable repair or long-term clinical benefit for the patient. In contrast to a comparator treatment, the microfracture, in which you drill a few holes into the underlying bone, it detects some of the subchondral bone marrow stem cells there, and you have some fibrocartilage repair. That repair can be fine in the short term, but in the long term, ultimately, it will be failing. Obviously, it was very difficult to define what is short term and what is long term. It was not very well known in the field. This is also negotiation with surgeons and the regulators, decided that at 12 months we were going to have the first outcome with a cartilage biopsy, a biopsy of the repair lesion. Long discussions on that, because you repair your cartilage and then you take a biopsy, but ultimately it was done, and the results were quite convincing in this sense, and this was published in the American Journal of Sports Medicine, that indeed implantation of this cartilage graft with expanded chondrocytes was more successful looking at the structural repair as analyzed by histology and of the microscopic biopsy of these patients here. Was it clinically relevant? These were the results after 3 years. Indeed, after 3 years, 36 months, there was a clear superiority of the cartilage transplantation than of the microfracture procedure, which here on the bottom, which became significant, I think, after 2 years and improved over time. So it was these results that ultimately give them the market approval in 2009. This is a historical date, June 26, 2009 in London, where the EMEA gave the first marketing authorization for an advanced therapy medicinal product. Was it all done and could we go to the market and start being happy and whatever? No, because then another challenge comes in, reimbursement. Who's going to get this treatment? Are we going to give this treatment to all patients with cartilage defects out there? That would be a lot of patients. Probably not many of them would sufficiently benefit from this relatively expensive treatment here. We are talking about 15,000 euro as a treatment here. So it was important to try to identify the patient at risk. Which are the patients that will really take advantage of this treatment and that will give a good clinical outcome here. And there is, I think, where for the ATMPs, when we talk about a living drug interacts with the environment, which is the patient. There's a crosstalk between these two. So when it's important to try to understand what is for a patient, what gives a patient a good chance to come up with a successful outcome of these treatments. And then the 5 years data were very instrumental. Here you see at the left-hand side the results after 5 years in the patients that all had a relatively recent symptomatic defect, and at the right-hand side were a number of patients that had a long-standing symptomatic cartilage defects for which eventually they already had a microfracture treatment which was not that successful and so on. And then you can see here, for instance, that this is one of the factors that seems to be critical if you want to have a successful repair or regeneration. I think it's a general principle, probably not only for the cartilage. If you want to have a good outcome of a regenerative treatment, it's better to enter early than to enter late in the game, because late in the game already many secondary changes have taken place that are affecting the joint and its environment. And here the results after the group of patients were quite significant after 2 years, 3 years, 4 years, 5 years. And this is where the benefit was the most convincing here. I see we are getting close to the end. My timer is telling me we are at 45 minutes. And indeed, we are perfectly in time. Thank you very much. And I'm passing on the thing again to our...
Thank you very much, Professor Luyten, and also thank you very much to Kristel. I think you give a wonderful presentation, and we also already received several very interesting questions. One of the questions is directed to Dr. Van Landuyt. So during reconstitution, how do you control the risk of contamination?
Well, I think that during reconstitution, it is good to take measures so that the product is not contaminated. Of course, in general, you will avoid all kinds of open steps. I think that this is a very important principle, that open steps are not necessary if possible. So yeah, what is an open step? That's another question then, of course. So the product has not to come into contact with air, with the environment. So it will depend on what you receive. And I think that the best way is that the manufacturer foresees the reconstitution, so makes a product and a packaging so that there is really minimal risk of contamination, knowing that indeed the regulation is not so strict about what environment it should be done. So it's really something that is seen as the bad side of the patient. And yeah, I think that's a very good remark, that for each product, the manufacturer and the user should make a risk-based approach to see what can be done. Of course, you can also do your steps of reconstitution in a controlled environment if you want to do that, but it's not prescribed by law that you have to do it in GMP environment.
Okay, thanks a lot, Kristel. So another question came on the short term and even on the long term more. Some collateral effects of ATMPs are still relatively unknown, so there are many unknowns. How can manufacturers and regulators work together to address these unknowns for these truly novel therapies? Professor Luyten, can you answer this question?
Yeah, I would say any novel treatment that enters into the patient with ATMPs should include long-term follow-up, so that at least a number of patients are followed for longer term, 5 years and even beyond. One could even think, depending on the type of products you have, that a really long-term follow-up up to 10 and 20 years will be required to know potentially what is happening with the transplanted cells, if they don't start walking around, especially if we talk then about starting from stem cells, if they are not starting to grow some strange tissue somewhere in the body. So pharmacovigilance is the terminology, is for these products is extremely important. With long-term follow-up I would say is for every drug development important, but for the ATMPs for sure, as there is still very little information out there. It is less of a concern in my view for autologous products, because essentially you are bringing back a cell in the patient from the young patient. Obviously, for allogeneic patients or allogeneic products, it becomes increasingly important for sure. And if we are starting to use what we call engineered cells, cells that have been somewhat changed in their original physiological functions, that will then require even more pharmacovigilance and follow-up of these products over a longer time period, that's for sure.
Okay. What is actually, in fact, custodianship, Dr. Van Landuyt?
Well, custodianship is the principle that human bodily material that is not a good that you trade like something that you produced. So it means that nobody can. It is a principle that says that the material is not owned by one person. I know that, of course, in all kinds of agreements, legally, sometimes it's spoken about I own the material and so on. So that is to solve certain judicial questions in certain circumstances. But for the general acting with this material, it's very important that as a group, for instance, different players, so doctors, manufacturers, biobankers, and so on, can be together responsible for the traceability, for the ethical way of working with this material. And so that is a framework that is very specific to human bodily material, and that will, in the end, has as aim to be responsible in a human manner, so that there will be no trade of bodies in the end, and that we will not have a stock market on body material in the end. So I think that most countries in the world would agree that there is a border on how to deal with this. And I think that custodianship is a nice way to call it by one word, this way of doing.
Okay. Professor Luyten, innovative manufacturing holds also a key to bring the prices of these very expensive ATMPs down. Sometimes, and we are asking now to you, what can we teach? How can we teach our ATMP students on the future focus of this problem of manufacturing, and what is the role of lowering down prices based on manufacturing optimizations?
Well, there are several aspects that need to be kept in mind. Number one is obviously the cost of a product is not only defined by its manufacturing cost. That's number one. That's a critical issue, and one would be surprised that it is lower. The manufacturing cost is lower than one anticipates. There is the investment in the clinical trials, multi-center trials, randomized trials, eventually duplicating trials, increasing the number of patients. So cost of manufacturing is only one part of the entire cost of a product. That's number one. But number two, okay, yes, for sure. If we could reduce the manufacturing cost, that would be already helpful. And I think the industrialization of the manufacturing is a process that has just started. In general, all these first products are still grown in what I would call 20th century lab technologies, which is technicians and gowns and laminar flows and incubators and these kind of things. I think the industrialization of cellular therapies is progressively starting to become a reality, and that includes a number of principles, being, for instance, miniaturization, which is reducing the sizes of things. And that is perfectly possible. Optimizing a relationship media over cell number. If you look at that, you can truly reduce the number of or the amount of volumes and media you need to handle. Number two, then closed systems. Instead of working in big places with complex ventilation systems and this and that, one could develop closed benchtop systems that where everything is happening inside, where things are automated, where there is no technician coming in, and this is then the robotic technologies. So these are the kind of steps that that are really ongoing right now, and that will start to reduce the cost of the product. But still, I think the cost of the product can also be reduced by then the volume. The more volume you can produce, the more patients that need your treatment, the better the price will become. So this is an additional aspect we'll recommend. If we keep having treatments of, we will treat 200 patients across Europe with 200 cell-based products, and these volumes are not allowing you to really reduce also the cost of the process. So, but it has many aspects. But I would say for the students, automation, robotics, closed systems, these are the kind of principles that they need to have in mind.
Yeah. Okay. Thanks a lot, Dr. Van Landuyt. During reconstitution, how do you control the risk of contamination, and are there any regulations for this?
Well, okay. Do you hear me? Because I was on mute. I was muted.
Yeah.
So the question is now the regulation for reconstitution activities, I think. Huh?
Indeed. And how do you control the risk of contamination during reconstitution?
Okay. I think that the risk of contamination, we already spoke about it in the previous question. The risk of contamination, but the regulation on it. Well, I think that if you want to speak about regulation, then it's the normal regulation for medicine, in fact, that is in place there. So the Utralex is not giving anything specific for ATMP reconstitution, except then the fact that there is a list of activities that are included, like I gave examples, like thawing. This is of course reconstitution, and everything that is more than that, which does not fall in it. So that's an important aspect of the regulation, that you know that what you're doing is in fact reconstitution and not something else. That is the most important. And when it's not reconstitution, it is GMP. So when it is reconstitution, it is just like the normal pharmaceutical, the normal pharmaceutical reconstitution that is also for other pharmaceutical products, like when you have to load it into other vials, or I mean, you have to make a solution, or so the regulation that is applicable for every medicine that is used in a clinic, and that has to be placed in a solution or anything.
Okay, thanks a lot. Professor Luyten, for Control Select, how long does it take from biopsy to implantation?
Hello, hello.
Yeah.
Yes, we hear you.
Do you hear me now? So again,
yes.
the totality is biopsy day 0 in the lab day 1, 3 weeks of expansion, delivery to the patient 1 day later, 2 days later, with a shelf life of about 48 hours. So it's a good 3 weeks in general.
Okay, thanks a lot. And how can you then prove that an ATMP is significant? I think the question is significantly effective at the clinical trial, since each product has its variability. So in an autologous sense,
well, yeah, for sure. But this is why you need to calculate the number of patients with clear outcomes, so that essentially the statistics is telling you that even with that variability, that the outcomes are still superior over the comparator. And it's one of the challenges, obviously. Yeah? So it's not a product which one can completely harmonize across all patients. So the product has an intrinsic variability. Therefore, the challenge of the clinical trials, obviously, that's for sure.
And some other questions come in. So for animal experiments, why isn't GMP not applicable? The cost is definitely a matter, but non-GMP conditions could also induce some variabilities in the experiment.
Yes and no. I would say for animal experiments, you have GLP, good laboratory practices, for instance, which means at the end of the day that your process is already quite robust and reproducible. That's one thing. But obviously, transition from GLP to GMP brings its challenges, and it's not the first product that had failed to do so and that lost its biological activity or the biological outcomes in patients. So from that perspective, indeed, one could request GMP manufacturing for animal experiments, but the bottom line is that the cost of developing a product would become very high, because GMP transition is a very expensive exercise here. So if you would already have to generate data in an animal with GMP manufacturing, with the risk that a lot of products will fail at that level, there will be very few people developing ATMPs going into the patient because of the incredibly high cost and just already to do the animal experiments. So there are indeed some limitations, and they are mostly related to cost and benefit.
Okay, thanks a lot. I think we are now already at 2 o'clock. Maybe I can give over the slide deck to Merel.
Hello. Yes. So thank you all for your time and attention. As I said in the beginning, the recording of this webinar will be made available in a couple of days on the website of EATRIS. You will also get an email with the recording and also the handouts. In 2 hours' time, you will get a follow-up email. Also having an evaluation form. So we would like to ask you to fill in this evaluation form. Your feedback is very valuable for us, also for improving our webinars in the next cycle of webinars next year. If you would have any other questions, please email them to advance@eatris.eu. And then I would like to welcome you already to next webinars planned after the summer, a technical webinar on pricing and reimbursement, and a career coaching webinar on stress and well-being. More info will be made available through our channels. Thank you very much and have a very nice afternoon. Bye-bye.